I'm Sajid Ali working at the University of Agriculture, Peshawar, Pakistan, and I'm working on the population biology of wheat rust. So I'm looking for wheat rust in the wheat fields, but as recently it's shown that yellow rust and other rust can go on the barberry species because it's the alternate host of wheat, contributing to the life cycle, making recombination cycle here, so you have more diversity if it is going on the barberry. So we are looking on the barberry. And secondly, our work has also shown that the center of diversity of wheat yellow rust is in the Himalayan region, so starting from Pakistan till India, Nepal and China. And barberry is a lot there, so some reports mention that there are more than 29 species there. So we are looking for to see the role of barberry in the epidemiology of wheat crop. And for that, to do that, we are looking for the acia on the barberry and the pycnia. What occurs that on the wheat crop we have the uridinospores, and the uridinospores then produce teleospore, and the teleospore produce basidium, which infect barberry. On the lower surface it produces the aeciospores, and these aeciospores can then go land on wheat and produce the next epidemic cycle. Now, it can receive rust sample from different grasses, so like Puccinia striformis, from poi, from dactylis and other grasses. So what we will be doing, we will collect the samples. We will collect these infected barberry leaves with acia. Now, with these samples there are two possibilities. The one is to do biological test, and for biological test you have to take the whole sample, and that whole sample you have to put in the glycine bags. These are the bags which are commonly used by the breeders during emasculation and crossing, but if you don't have these bags you can simply use paper bags. You put the leaves in this, and for biological tests we take more number of leaves, the better it is. Then you would like to make the proper code of the sample. So like here I'm in Nepal and I'm in a location, so I give this location 1 and this is the sample 1. I put the date, right? And then in an Excel file, this code NP11 will have a specific meaning with which I will write the name of the place, the date, the GPS data, the infection severity on the barberry plant, and any other information which could be used later on. And that file will go with the samples. Now, for the living samples, it could be processed at the Eugene's lab at USA. So you can send these samples to them, and they have special quarantine facilities where they can use it. In the biological test, they have a panel of grasses and wheat susceptible lines on which they will inoculate the acia from these samples, and with that we can see if it is going on wheat or any other grasses, and that will then tell us that this barberry plant was infected by which rust and which formis specialis of that rust, Puccinia graminis triticii, Puccinia striformis triticii, or Puccinia striformis formis specialis poi. This is the one step. Another way to do that is to have the samples for molecular work. So with the sequencing techniques we can have an infected lesion, and that infected lesion we can extract the DNA from that, and once the DNA is extracted we can use molecular markers and then sequencing, and when we compare the sequencing of this infected acia with the grass rust or the wheat rust, we can tell if it is Puccinia striformis, Puccinia graminis or any other related species. That can be done again at the Eugene's lab, so you can send the samples to him, or I can also do that at Pakistan in collaboration with Global Rust Reference Center, Denmark. For DNA samples, you detach the leaf with acia, and then you just cut the leaf around the acia, and you just leave this aecial cluster which contains several aecial spores. So now you have the lesion, and you can use these cryotubes But if you don't have these cryotubes, you can use a simple 2 ml Eppendorf tubes. So you put this lesion to this cryotube or Eppendorf tube and then put some alcohol to this sample. Cover it and leave it for 2 to 3 days. The spores, the acia and any other organism or microorganism on the leaf will die with this, and then after 3 days you just throw out the alcohol and send this dry sample either to my lab or to Eugen lab having their consent, and then we will extract the DNA and with molecular markers with sequencing technique we can tell which species and Formis specialis was on this sample. So sending the sample from this region to any of international lab or even in the region to another lab, it's better to have this alcohol technique because this will kill the pathogen and it will not go outside the lab in those countries and infect the wheat crop. So that's why we put ethanol alcohol here, and that will avoid any contamination. Once you have the sample, you have to coat properly this one, and this coat could be the same as I did, like Nepal 1 sample 2, and that should then correspond in the Excel file to specific information. So like plant number, the plant species if you could know, and you can take the help from a botanist to identify the species. You can put the date, you can put other information, whether it's prolifically infected or it's just very minor infections, whether it's close to a wheat field or it's away from a wheat. And you can also, from the date, of course, you can add whether it's the crop is still there, at what stage. So that will explain the real role of this infected barberry to the epidemics on wheat. Another important point is in one cryo you try to put only one aecial cluster, because the two aecial cluster on the same leaf could be different genotypes. So we would like to have only single aecial cluster. It's not like the living sample where you would like to have everything, and then in the greenhouse and in the lab you would like to have all the infection and do monospore culturing. In this case we would like to have a single cluster and then do the further sequencing. In literature we know that the basidiospores and the aeciospores, they could not travel the long distance as the eurydiospores can. So a distance of 5 kilometers till 5 kilometers from wheat crop will be okay, and beyond that, below this is preferable, and beyond that you can skip. At what time? So the time is really important in this context, because we don't know at what stage the acia can go from here to the wheat, or the talia can come on barberry, the basidiospores can come on barberry. So what we can do, we can have the same tree, we identify the tree and we do a sampling in January. If it is an evergreen barberry, we do in March, then we do in May, then in July, and we can do with 1 month interval. And that information should be written in the file you are sharing with the concerned lab, so that we know that it's the same barberry bush with different interval time. Then you can also have different barberry bushes in the same way, and you can do it in a specific location. You can go around that particular location and do, like in one location, 10 barberry bushes, but they should be distant and representative of the location. Now, concerning from which country we can receive the samples, so as I mentioned, for the DNA samples, the samples are already dead, so we can receive it from the quarantine aspects. We can receive the sample from anywhere. The same for the living samples. At the Eugen lab they have quarantine facilities, so they can receive from any country. But considering the information on the center of diversity and the level of diversity of Pakistania, we would like to receive the samples from anywhere in the Himalayan and near Himalayan region, like Afghanistan, Pakistan, Nepal, India, Bhutan, and anywhere in this region if we have barberry. So we would be interested to see that, and of course the information and the data would be shared with the concerned country. Finally, I would emphasize on the contribution of international community to understanding of rust biology at worldwide level, what is known from the time of Stubbs and then our collection and the future one. Without your collaboration, we couldn't understand this population biology of rust pathogen, and in collaboration we can really cope with the challenge and with the problem of rust. Thank you for your collaboration, and thank you for watching the video.